We have been talking about GAGs, which are basically molecules made up of carbohydrates. One of the things people get confused about is proteoglycans and glycoproteins. You would come across these two terms fairly frequently in literature if you're studying cell biology or molecular biology. So let me straighten out the record and explain what is the difference between these two. Number one. Number two, unfortunately, there are a lot of these molecules that I'll be talking about. I will like you to pay specific attention to the ones which I talk about with the help of a diagram. So I realize memorizing all these molecules could be difficult, but at least please remember the name of molecules and their features, especially the ones which are represented graphically. So first of all, let's get our record straight and see what is the difference between proteoglycans and glycoproteins. Glycoproteins contain 1 to 60% carbohydrate by weight in form of numerous, relatively short, branched oligosaccharides. So special attention to numerous, relatively short, branched oligosaccharides. Okay, so we'll see how it contrasts with the proteoglycans. They're usually not branched carbohydrate molecules. Proteoglycans, they contain more carbohydrate material by weight. They can contain as much as 95% of the total dry weight, and they generally form long, unbranched GAG chains. Each is typically about 80 sugars long, and at least one of the sugar side chain of proteoglycan must be a GAG. So this is basically the difference between proteoglycan and glycoproteins. Both are basically made up of protein and sugar molecules, but terminology, the way we define these two molecules is that, number one, glycoproteins have less carbohydrate as compared to proteoglycans, which is up to 95%, and proteoglycans have to meet this condition that at least one of their side chain has to be GAG, and also they are not generally branched. Proteoglycan, let me give you an example of proteoglycan, an aggrecan molecule. I'll show you a diagram of aggrecan. It's an important molecule. You should learn more about this particular molecule. I'll test your knowledge on this molecule during the exam. Aggrecan is a major component of cartilage, and it is about the size of a bacteria actually. Its mass is 3 into 10 raised power 6 daltons. It's 10 raised power 6, and it is over 100 GAG chains, which are basically chondroitin sulfate, and about 30 keratin sulfate molecules are attached to the serine-rich core protein of almost 3,000 amino acids. So the core protein is 3,000 amino acids, and it is attached to 100 GAG chains, and also out of 100, there are about 70 of these chondroitin sulfate chains and approximately 30 keratin sulfate chains. We'll see the structure of this molecule later on, but it's a fairly large molecule. Let's look at basically some other proteoglycans. Here, for example, it's decurin. Here it's shown it has only one GAG. So by definition, it is a proteoglycan, not a glycoprotein, because it is attached to one carbohydrate chain, and that is a GAG. Here is aggrecan. We talked about this in the previous slide. Here is a core protein, and out of these core proteins, you have carbohydrates attached to it. Here is actually a molecule which is a glycoprotein rather than proteoglycan. If you look at the slide more carefully, you will see that this sugar, this red structure here, is branched. I'll draw it out in a magnified form, and it is attached to a protein which is the polypeptide chain. So it is short, branched oligosaccharide. This is basically structure of ribonuclease. It has a molecular weight of about 15,000. So basically, we are looking at proteoglycans compared with glycoproteins. So this protein, I would like to mention decurin. Basically, it's called decurin because it decorates the fibrils of collagen 1, and it is important for the functionality of collagen, and it is secreted by fibroblasts and has a single GAG chain, as you can see, as I pointed out. Pancreatic ribonuclease B, I've already talked about this. It's also a secreted glycoprotein, but it is a glycoprotein, and these two molecules I've shown are the proteoglycans. Except hyaluronic acid, which we have talked about in the previous module, all GAGs are covalently attached to protein in form of proteoglycans, which are made by most animal cells. So as we have talked about earlier, that the ribosomes, which start to make a protein which has a special sequence, these ribosomes stop making that protein and they first dock on the surface of ER, and then the protein synthesis resumes, and protein is co-translationally inserted into the lumen of ER. Here, this protein goes through certain modifications. We have talked about this process. I don't want to take too much time on it, but all the way from ER to Golgi, sugar molecules will keep on getting attached to the core protein, and it will get decorated with these carbohydrate molecules. So polypeptide chain or core protein of a proteoglycan, again, is made on membrane-bound ribosomes and threaded into the lumen of endoplasmic reticulum. Now let's see how these sugar molecules are attached to the protein. First of all, let me point out that in majority of the cases, these modifications take place on the serine residue. First of all, let me point out that these modifications take place on serine residue. The way it starts is that a tetrasaccharide, which basically means 4 sugar molecules attached to each other, and those sugar molecules are xylose, galactose, and glucuronic acid. So it's basically this is a tetrasaccharide, 4 sugar molecules attached to each other through glycosidic linkages shown here by this line. These first of all get attached en bloc to the serine residue in a polypeptide chain. Basically, serine is one of the amino acids, and since this amino acid is part of a polypeptide chain, so that's why I'm referring to as a residue rather than an amino acid. So first of all, again, these 4 sugar molecules get attached to serine. Which serine is selected? A polypeptide chain can have several serines. Not all serines are glycosylated, or they are not attached to sugar molecules. It turns out it's not just the sequence, that there's a particular sequence of amino acids in amino acid residues in a polypeptide that are selected. It is basically the shape or the serine that is forming a particular structure. It is a structure that is recognized rather than the sequence of the amino acids. So these, first of all, 4 sugar molecules that are attached to this serine en bloc, and then later on, these GAG molecules are attached. Here we are seeing A and B. So these molecules are attached one monomer at a time to the seed of this 4 tetrasaccharides. So the seed is basically where this additional sugar molecules will be attached, and it will grow from this particular seed. So this process is basically catalyzed by glycosyl transferase, and it, again, as I've mentioned, looks for a local conformation rather than the sequence of amino acids. The core proteins can vary greatly in number and types of attached GAG chains. The underlying repeating pattern of disaccharide in each GAG can be modified by a complex pattern of sulfate groups. We have already talked about how some of these GAGs have sulfate groups attached to the sugar molecules. So there are further modifications of even the sugar molecules that have been attached and are part of GAG. This heterogeneity makes it difficult to identify and classify proteoglycans in terms of their sugar sequences of core protein, because the sugar sequences and core proteins are extremely diverse. Proteoglycans is a diverse group of highly glycosylated proteins whose functions are mediated by both their core protein and, very importantly, the GAG chain, which is attached to these core proteins. So function of this protein is basically dictated by 2 entities, the protein and, of course, the sugar molecules that are attached to it.